It's my pleasure to introduce Carolyn Coussens. She's a second-year veterinary student at Mississippi State University with a special interest in pathology, parasitology, and invertebrate medicine. She hopes to pursue a pathology residency after vet school. She is known among her peers for her odd choice in pets, as she currently owns and cares for 9 Chinese praying mantises. That's an impressive number. All right, thanks, Carolyn, and you can take it over from here.
All right. Can everybody hear me?
Yep, sure can.
All right. Good afternoon, everybody. My name is Caroline Coussens. So I worked on this project as part of the Summer Research Experience program at Mississippi State University's College of Veterinary Medicine. So today I'm going to be talking about Diplostomid Trematode life cycles and how my summer research experience focused on the life cycle of one particular trematode called Hysteromorpha. So before we get started with anything, I'd like to give a general overview of the trematode life cycle. Let's see. Yeah, there we go. So trematodes, more commonly known as flukes, are a group of monoecious parasitic flatworms that make use of an indirect life cycle. So that's basically where different host species are parasitized by distinct life stages of a single trematode species. The general life cycle for catfish typically involves eggs hatching into Miracidia, which then go on to infect a snail first intermediate host. These snails shed cercaria that then encyst as metacercaria in a fish second intermediate host. The life cycle is completed when these infected fish are consumed by a piscivorous bird definitive host, with the adult trematodes residing in the bird's GI tract. Understanding which trematodes are in which host is important because interrupting the trematode life cycle is often the only avenue we have for preventing trematode infections. There are a lot of food production animals that currently have no approved anti-parasitic medications. Take, for example, commercial catfish aquaculture. So there are no approved antiparasitics for catfish, so producers have to rely on identifying and eliminating host snails from their ponds if they want to prevent or treat a trematode infection. And there's a lot at stake here because to date, the commercial catfish industry is the largest aquaculture industry in the United States. So according to the USDA, it was worth an estimated $360 million as of 2018. Most of the industry is concentrated in the southeastern US in states like Louisiana, Arkansas, and Mississippi alone accounts for over half of that production. Current challenges facing the catfish industry include the recent rise in foreign imports competing with local production. Approximately half of all catfish morbidities and mortalities are attributable to infectious diseases, which includes those that are related to trematodes. And then the last big problem is the consumption of catfish by piscivorous birds. So these would be your egrets, your herons, the American white pelican, and one of the most damaging is this little guy, the double-crested cormorant. So not only do these guys consume an obscene amount of fish relative to their body weight, but they're also the definitive host for multiple trematode species that infect catfish. These species include Ostradiplospomum compactum, Trypanocephalus spathans, Riboroia, and today's topic of discussion, Hysteromorpha. So Hysteromorpha is a diplostomid trematode of catfish that is characterized by its spatulate anterior end, tapered posterior end, and its prominent tribacidic or holdfast organ, which is typically used for attachment. So at the time I started this project, we still didn't really know for sure what Hysteromorpha's first intermediate snail host was. Historically, that hasn't been much of an issue or priority in finding out because it was thought that even though Hysteromorpha could infect catfish, it wasn't responsible for any significant pathology. However, in the summer of 2019, the Rosser lab saw to a case of reduced feeding activity at a commercial catfish operation in the Mississippi Delta region. And when these inappetent catfish were necropsied, they were all found to be heavily infected with, take a guess, Hysteromorpha. So you see, the thing with Hysteromorpha is that when it infects catfish, it insists very deep in the musculature. Compared to other trematodes, like say Bulbophoris, with Bulbophoris you can see lesions on a fish and know that it's infected because they insist near the surface. With Hysteromorpha, you can't tell whether a fish is infected just by looking at it. So there's a concern here that the way Hysteromorpha insists could be obscuring Hysteromorpha attributable losses, or that producers will invest all of this time and money into raising infected catfish, only for the fillets to get rejected at processing because they're laden with all these metacercaria. If we knew Hysteromorpha's life cycle, producers could just target the snail host and prevent the infections outright, and it would also have the side benefit of clearing up some of the taxonomic confusion around Hysteromorpha, because as it currently stands, Hysteromorpha literature is kind of a mess. The species was originally described as Dystoma trilobum in 1819, and it cycled through a whole bunch of different names before consensus finally settled on Hysteromorpha triloba. Hysteromorpha triloba was considered to be the only species in its genus until 2018, when Sean Locke's group took a look at the molecular data and concluded that the North American Hysteromorpha specimens were genetically distinct enough from the European specimens to warrant their own species called Hysteromorpha corti. And on top of that, there's also a third suspected species in South America. Long story short, it's pretty complicated right now, and as a result, a lot of these major genetic sequencing databases like GenBank likely have their North American Hysteromorpha corti sequences misidentified as the European Hysteromorpha triloba. Completing the life cycle will allow us to compare Hysteromorpha sequences and figure out which species is actually present in Mississippi. So with all that in mind, these were our objectives. We wanted to molecularly and morphologically identify different Hysteromorpha life stages in order to complete the life cycle. And we also wanted to find some Hysteromorpha Cercaria to challenge channel catfish fingerlings with in order to determine the infectivity of the Cercaria and whether there was any resulting pathology. So here's how we did that. So first, we screened the small intestinal contents of 26 double-crested cormorants for adult Hysteromorpha. We stained these adult specimens with Van Cleve's hematoxylin and then did a qualitative analysis of the morphology just to make sure that they fell in line with Hysteromorpha's type morphology. We also stained and assessed Metacercaria that were collected from fish back from that original Hysteromorpha outbreak that was noted by the Rosser lab in 2019. Next, we collected snails. We went to commercial catfish operations in Sunflower and Noxubee counties in Mississippi, collected all the snails we could find, which added up to about 3,000 by the end of the summer. We tubed each of these snails individually and then screened them at 24 and 48 hours each for the presence of shedding Cercaria with Hysteromorpha type morphology. So Hysteromorpha Cercaria typically look like this. The most important characteristic for identifying Hysteromorpha is the presence of a ventral sucker, which is found near its anterior end. So we took a subsample of these adult Cercaria and Metacercaria that we'd found, and then we used them for molecular analysis. It's important to pair molecular data with morphological data when you're doing taxonomic work, because oftentimes these trematodes that look very similar morphologically look drastically different genetically. We amplified the DNA from our specimens using PCR and compared their sequences to other Hysteromorpha sequences that were available in GenBank. For this project, we chose to sequence the cytochrome C oxidase mitochondrial subunit gene because it's highly conserved between species. And we also took a look at the internal transcribed spacer region and 28S ribosomal subunit genes because those have historically also been used to do taxonomic work with trematodes. These trees would help us identify what Hysteromorpha species was present in Mississippi, as well as confirm that each life stage we recovered was in fact the same species in order to complete our life cycle. So lastly, we wanted to use some of the Cercaria with Hysteromorpha type morphology that we found and challenge channel catfish fingerlings with them. We made use of 28 experimental pathogen-free channel catfish fingerlings, which were housed in individual cups with air stones to provide oxygen to them. We also set aside 20 fingerlings as negative controls, which were then housed in a group setting. We plan to expose each of these experimental fingerlings to 200 Cercaria, and then necropsy 2 experimental fish and 2 negative controls every 10 days post-infection in order to look for evidence of Metacercaria growth. So after doing all of that, these were our results. So before we start talking about the phylogenetic stuff, I just want to show you the results of our double-crested cormorant small intestinal survey. I especially want to emphasize that the unequal distribution of parasites among these birds, only 32% of the cormorants we necropsied were actually positive for adult Hysteromorpha. And the intensity of the infection among those positive birds ranged anywhere from 1 adult to 77 adult specimens, with a mean intensity of 12.3. Other species like Echinocasmus had an even wider range of infection intensities. So the main thing I want to take away from this is that just because a species is listed as the host for a given parasite, that doesn't mean that every single member of the species is going to have that parasite. You only need 1 heavily infected cormorant going in for a swim in order to infect an entire catfish pond. So there's my soapbox moment for the day. It only takes 1, folks. Let's see. Moving on to phylogenetics, here we have a tree for the COX-1 mitochondrial subunit gene. So at the bottom of this tree, we have some non-Hysteromorpha diplostomid species, such as Diplostomum and Ostrodiplostomum, and these serve as our outgroup. As we move up, we see 3 Hysteromorpha specimens from South America. These are that new species from South America that I mentioned earlier. Next, we have a species from Europe that has been confirmed to be Hysteromorpha triloba. And then all of these, this big bracket here, is North American Hysteromorpha sequences. So these include all of our sequences, which are highlighted in yellow. And then it also contains right here a species that is a sequence that has been confirmed to be Hysteromorpha corti. So because all of these North American specimens are so genetically similar to each other and are clearly separated from both the South American Hysteromorpha species and the European Hysteromorpha triloba, we believe that our sequences are in fact a separate North American species, Hysteromorpha corti, and that all of these that are labeled Hysteromorpha triloba here are just mislabeled in GenBank. Because keep in mind, a lot of these are older than 2018, and 2018 was when they first looked at the genetic data and decided that it was a distinct species. So the fact that all of our sequence life stages place so close to each other on the tree also supports the idea that each of these stages belong to the same species. All of our stages were like more than 96% similar, which warrants them being the same species. And when we look at the trees from the 28S and ITS regions, even though these are less specific than our COX-1 data as far as species specificity goes, they still support the idea that all of these stages are conspecific. So if you look at these red boxes here, you'll notice that we only used a single Cercaria specimen in these trees. So this sequence came from an archived Cercaria specimen that was collected in the Mississippi Delta region that was suspected to be and then later confirmed to be Hysteromorpha corti with genetic analysis. So these Cercaria were shed from a planorbid snail, which is more commonly known as a ram's horn, which implicated the ram's horn snail as the first intermediate host for Hysteromorpha corti. By contrast, do y'all remember those 3,000 snails we collected over like the course of a whole summer and checked all of them twice? Well, guess what? We only found 3 ram's horn snails that were shedding Cercaria that fit the type morphology for Hysteromorpha. So after all that searching, we're like, thank God, the morphology matches, has the ventral sucker, we finally found them. So we set the fish challenges up, sent off the sequence data, got the sequence data back. And remember how I mentioned earlier that you can't rely on morphology alone when you're doing taxonomy? Y'all, guess what? They weren't Hysteromorpha. This is what I get for my hubris. So not only are they not Hysteromorpha, they don't match any of the sequences that are available in GenBank. So that means we either have molecular data for a known species that has not yet been sequenced, or these Cercaria here are a completely novel species. So that's where this current project sits. Just to reiterate the high points, the Diplostomid Cercaria we found showed us just how unreliable morphology is on its own and why you need molecular data in order to do taxonomic work. We kind of tricked ourselves there by relying on the morphology. When we looked at that molecular data, our phylogenetic trees supported the idea that 3 separate Hysteromorpha species existed. There's Hysteromorpha triloba in Europe, there's Hysteromorpha corti in North America, and then there's a 3rd Hysteromorpha species in South America. We found it particularly interesting that the South American species was less genetically similar to Hysteromorpha corti than Hysteromorpha triloba was, considering that cormorants in North America and South America have ranges that overlap quite a bit. So it makes us wonder what else is influencing the distribution of these parasites, aside from just like their host species range. Our molecular data also supports the idea that our Hysteromorpha specimens were conspecific, and we've identified the first intermediate snail host for Hysteromorpha corti as the ram's horn snail, thus completing the life cycle. So we can edit this list of catfish trematodes that use double-crested cormorants as their definitive host to include Hysteromorpha corti instead of Hysteromorpha triloba. So in the end, we answered the question of Hysteromorpha's life cycle, but we still have a lot of work left to do. First and foremost, we still have one of our objectives to complete. We still need to find some actual Hysteromorpha Cercaria to do our challenges with, because even if we've identified the snail host, the usefulness of that information is going to depend on how much of an impact Hysteromorpha has on catfish morbidity and mortality. We also need to identify the Diplostomid Cercaria we found. There's quite literally dozens of Diplostomid species that haven't been genetically sequenced yet, so we have our work cut out for us. Our current guess is that it's some kind of Alaria species, since that's what its closest match was to in GenBank. Also, in addition, when we were collecting all of those snails by looking at Hysteromorpha Cercaria, we also accidentally completed the life cycle of another common trematode in catfish ponds called Elucidation, and we found at least one species that uses catfish as its definitive host. So we're currently in the process of sequencing a bunch of Elucidation Cercaria to see just how many species are present in Mississippi catfish ponds and which ones infect catfish. In addition to that, we also identified a snail host for another trematode that was found in cormorants called Echinocasmus. I swear I spent so long looking for these Hysteromorpha and I found everything else instead. So this is Cincinnatia integra, and it's found in the northern half of Mississippi and Alabama. So compared to ram's horn and biomphalaria, these snails are found a bit farther out in the catfish ponds. They seem to congregate in finer sediment and instead of hiding under aquatic vegetation like ram's horns do. And there's evidence that heavy echinochasmus infections can cause pathology in catfish, so we also want to return to where we found these snails, collect some more Cercaria in order to do fish challenges with those as well. So all in all, I think it was a pretty busy summer. Buckle up, I have a lot of people to thank. Thank you all so much for listening. I'd like to thank Dr. Graham Rosser for his mentorship throughout this whole project and for allowing me to participate wholeheartedly in this lab. I wasn't just confined to Hysteromorpha, I could work on whatever I wanted, whenever I wanted. I'd like to thank Ethan Woolyard for teaching me how the lab operates, helping with the sequencing and the necropsies, and for helping me to just an ungodly amount of snails. I'd like to thank Dr. David Wise, Dr. Matt Griffin, and the Thad Cochran National Warm Water Aquaculture Lab for allowing us to use their facilities for our fish challenges. I'd like to thank Mackenzie Gunn for providing the Cercaria that allowed me to complete this life cycle. I'd like to thank Tommy King for procuring the cormorants that we necropsied, Jonah Wen and Lauren Easter for helping us necropsy those cormorants, Brad Richardson for helping with the catfish necropsies, Lori Ford for her occasional help in the lab and for being an all-around cool lady, and the laboratory praying mantises for being such great moral support to me. So I'd also like to thank Boehringer Ingelheim, the NIH, the National Wildlife Research Center, and the USDA Agricultural Research Service for funding this project. And lastly, I'd like to thank the Mississippi State College of Veterinary Medicine and this year's SRE mentors for allowing me to participate in their summer research experience. And with that, I will take any questions.
All right, thank you, Caroline. That was an awesome talk and impressive project for the summertime. We've got plenty of time for Q&A. If anybody has questions, let's go ahead and unmute yourself, or you can put it in the chat box and I can read it to her.
Hey, Caroline, this is Jan. I'll ask a question.
Hey.
You've got this nice soup of different intermediate hosts and definitive hosts in these catfish ponds. And you talk about how identifying the snail host is important for treatment. What sort of approach do they use in catfish ponds to treat snails to reduce their populations? And are there different treatments for different snail species? Like, what's the diversity of species in a pond? And would you use one approach if you know it's one snail species versus another snail species that's the intermediate host for the parasite?
Yeah, so unfortunately, there's not a lot of treatment options when it comes to eliminating the snails. The main treatment right now is like copper in the water because it's toxic to them. That doesn't really discriminate between snails. Copper will pretty much get rid of all of them. The problem is it doesn't just stress out the snails. It harms pretty much everything in the pond to a certain degree. And so it's kind of used as like the big guns. So the importance of the snail is mostly to like estimate the threat of certain infections in your fish. Like if you have a problem with like a certain parasite in your ponds historically, but you know that there are those snails that are the host are only in these certain ponds, then you only need to treat those specific ponds that have the snails. And so you can save your money and your effort trying to like, as opposed to like trying to like blast all of the ponds with copper, so.
Sure, yeah, that makes sense. Thanks.
Mm-hmm.
Any other questions out there?
I'll ask a question as well. This is Alex Primus. Very nice talk. Very interesting work. So I don't know a ton about treatment options and what's actually possible. I mean, there obviously are drugs to treat flatworms, not legally approved in the U.S. But do you know, if that could move through the legal process, would that be an option for catfish farmers, or is it just not? It doesn't make sense financially or for other logistical reasons?
I don't know too much about the specifics of that since we're actually pretty far away from what I can understand from having any medications approved. It would all depend on cost. I'm sorry.
Okay, no problem. Thanks.
All right, if we don't have any more questions, we can shift gears to the next presentation.